disease epidemiology. From the time that the first farmers began to cultivate plants, people have been concerned with reducing the crop losses caused by diseases. Through the evolution of our crop production technologies over the last 10,000 years, the principles of plant disease management have been woven into the fabric of our civilization. The decisions of when, where, and what to plant and the development of specific cultural practices have been based on countless generations of trial and error. Without a doubt, the successful farming methods depended upon being able to suppress the development of plant pathogens, even if the farmer had no particular awareness of the underlying biological mechanisms that led to her or his success. Everywhere in the world, either directly or indirectly, our attempts to manage plant diseases have determined what we eat and where we have settled. Plant disease epidemiology is the study of disease in plant populations. Much like diseases of humans and other animals, plant diseases occur due to pathogens such as bacteria, viruses, fungi, oomycetes, nematodes, phytoplasmas, protozoa and parasitic plants. Plant disease epidemiologists strive for an understanding of the cause and effects of disease and development strategies to intervene in situations where crop losses may occur. Typically, successful intervention will lead to a low enough level of disease to be acceptable, depending upon the value of the crop. Plant disease epidemiology is often looked at from a multidisciplinary approach, requiring biological, statistical, agronomic and ecological perspectives. Biology is necessary for understanding the pathogen and its life cycle. It is also necessary for understanding the physiology of the crop and how the pathogen is adversely affecting it. Agronomic practices often influence disease incidence for the better or for worse. Ecological influences are numerous. Native species of plants may serve as reservoirs for pathogens that cause disease in crops. Statistical models are often applied in order to summarize and describe the complexity of plant disease epidemiology so that disease process can be more readily understood. For example, comparisons between patterns of disease progress for different diseases, cultivars, management strategies or environmental settings can help in determining how plant diseases may best be managed. Policy can be influential in occurring of diseases through actions such as restrictions on imports from sources where a disease occurs. In 1963, J. E. van der Planck published Plant Diseases, Epidemics and Control, a seminal work that created a theoretical framework for the study of epidemiology of plant diseases. This book provides a theoretical framework based on experiments in many different host pathogen systems and moved the study of plant disease epidemiology forward rapidly, especially for fungal foliar pathogens. Using this framework, we can now model and determine thresholds for epidemics that take place in a homogeneous environment such as a monocultural crop field. Elements of an epidemic. Disease epidemics in plants can cause huge losses in yield of crops as well, threatening to wipe out an entire species such as was the case with Dutch elm disease and could occur with sudden oak death. An epidemic of potato leaf blight caused by Phytophthora infestans led to the Great Irish Famine and the loss of many lives. Commonly, the elements of an epidemic are referred to as the disease triangle, that is, a susceptible host, pathogen and a conductive environment. For a disease to occur, all three of these must be present. Below is an illustration of this point where one side of the triangle shows each of the factors. Where all three items met, then there is a disease. The fourth element missing from the triangle for an epidemic to occur is time. As long as all three of these elements are present, disease can initiate. An epidemic will only ensure if all three continue to be present. Any one of the three might be removed from the equation though. The host might outgrow susceptibility, as with high temperature adult plant resistance. The environmental changes and is not conducive for the pathogen to cause disease, or the pathogen is controlled through a fungicide application, for instance. Sometimes a fourth factor of time is added as the time at which a particular infection occurs, and the length of the time conditions remain viable for that infection can also play an important role in epidemics. The age of the plant species can also play a role, as certain species change in their levels of disease resistance as they mature, a process known as ontogenic resistance. If all of the criteria are not met, such as a susceptible host and pathogen are present but the environment is not conducive to the pathogen infecting and causing disease, disease cannot occur. For example, corn is planted into a field with corn residue that has the fungus Sericospora zeamalidis, the causal agent of grey leaf spot of corn. But if the weather is too dry and there is no leaf wetness, the spores of the fungus in the residue cannot germinate and initiate infection. Likewise, it stands to a reason if the host is susceptible and the environment favors the development of disease, but the pathogen is not present there is no disease. Taking the example above, the corn is planted into a plowed field where there is no corn residue with the fungus. Sericospora zeamalidis, the causal agent of grey leaf spot of corn, present but the weather means long periods of leaf wetness, there is no infection initiated. Let us now look at the types of epidemics. Monocyclic epidemics are caused by pathogens with a low birth rate and death rate, meaning they only have one infection cycle per season. They are typical of soil-borne diseases such as fusarium wilt or flax. Polycyclic epidemics are caused by pathogens capable of several infection cycles a season. These are most often caused by airborne diseases such as powdery mildew. Bimodal polycyclic epidemics can also occur. For example, brown rot of stone fruits and the blossoms and the fruits that are infected at different times. For some diseases, it is important to consider the disease occurrence over several growing seasons, especially if growing the crops in monoculture year after year or growing perennial plants. Such conditions can mean that the inoculum produced in one season can be carried over to the next, leading to a build of an inoculum over the years. In the tropics, there are no clear-cut breaks between growing seasons as there are in temperate regions, and this can lead to the accumulation of inoculum. Epidemics that occur under these conditions are referred to as polytic epidemics and can be caused by both monocyclic and polycyclic pathogens. Apple powdery mildew is an example of a polytic epidemic caused by a polycyclic pathogen, and Dutch elm disease, a polytic epidemic caused by a monocyclic pathogen. The cyclical nature of plant disease. Plant disease epidemics are cyclical phenomena, that is, they consist of repeated cycles of pathogen development in relation to the host and the environment. The inoculum, which might consist of fungal spores, bacterial cells, nematodes, viruses within an aphid vector or some other propagules of pathogen, gains entry into an establishment within the host tissues through the process of infection. The pathogen develops within the host and eventually begins to produce new inoculum, which in turn can be dispersed to new susceptible sites to initiate new infections. Pathogens that produce only one cycle of development, that is one infection cycle per crop cycle, are called monocyclic, while pathogens that produce more than one infection cycle per crop, like told before, are called polycyclic. Generally, in temperate climates, there is only one crop cycle per year. So, the terms monocyclic and polycyclic are based on the number of cycles per year. In tropical or subtropical climates, however, there can be more than one crop cycle per year, and it is important to remember that monocyclic and polycyclic are based on a single crop cycle. These same terms are used to describe the epidemics as well as the pathogens. So, we often speak of a monocyclic epidemic or a polycyclic epidemic. For some diseases, it is important to consider an epidemic over a period of many growing seasons. This is particularly true for perennial plants, which are foragers, pastures, lawns, orchids, forests, etc., or for annual crops that are grown in monoculture year after year. In these situations, the inoculum produced in one growing season carries over to the next, and there could actually be a build-up of inoculum over a period of years. In the tropics, there may not be clear-cut breaks between growing seasons, such as we find in the temperate zones, and epidemics can virtually be continuous over periods of many years on such crops such as banana, coffee and rubber trees. We refer to these epidemics as polytic epidemics, regardless of whether the pathogen is monocyclic or polycyclic within each season. Let us now look at the Dutch elm disease, which is a vascular wilt disease. The earliest external symptoms of infection are often yellowing and wilting, which is also known as flagging, of leaves on individual branches. These leaves often turn brown and curl up as the branches die, and eventually the leaves may drop off. Although initially only a part of the tree crown may be affected, symptoms may progress rapidly throughout the crown. High susceptible trees often die in a single year, but others may linger for several years. Symptoms progress quickly and the death may occur rapidly in trees infected in early spring, while trees infected later in the summer may survive longer. If the bark of the infected elm twigs or branches is peeled back, brown discoloration is seen in the outer layer of wood. This discoloration in the xylem actually occurs before the foliar symptoms described are seen. Foliar symptoms result when sap flow ceases in the infected wood. Xylem browning is often discontinuous. In cross section, it appears as a circle or brown dots or a ring. Other wilt diseases of elm, such as verticillium wilt, also cause sapwood discoloration. So, positive diagnosis of Dutch elm disease depends on laboratory culturing and identification of the fungus. This is an example of a monocyclic pathogen giving rise to a polytic epidemic. Note that while there is only one cycle of infection each year and the disease progresses within each year is roughly linear, the incidence of infected trees increases at an increasing rate from year to year. Apple powdery mildew is an example of a polytic epidemic caused by a polycyclic pathogen. Note how the incidence of infection at the start of each year tends to increase exponentially. Sigatoka leaf spot on bananas decreases somewhat during the dry season, but otherwise produces more or less continuously repeated cycles of infection. The host in this case consists of a population of plants of different ages developing continuously over a long period of time. Let us now look at relating disease progress to disease cycles. If we analyze the disease cycles of the epidemics for which we have plotted disease progress, one can see that the epidemics that progress roughly linearly or are linear at the start tend to be monocyclic epidemics. On the other hand, the disease that increase at an increasing rate during the early part of the epidemic tend to be polycyclic epidemics. Let us now look at in detail about monocyclic epidemics. In general, there are three types of plant diseases that tend to produce only one infection cycle per host. The first is the post-harvest diseases. The second is diseases caused by soil-borne plant pathogens. And the third is rusts without a uridinospore stage. Post-harvest diseases. Not all post-harvest disease produce monocyclic epidemics, but in many cases the infections that result in storage rots have either already occurred before harvest or occurred during the harvest and post-harvest handling before the product goes into storage. The rot progresses during storage and new inoculum may be continuously produced, but unless the stored product is handled again to disperse the inoculum and create new infection sites by making small wounds, there are no new infection cycles. Note that the pathogen is not inherently monocyclic but is constrained by the environment to produce only a single cycle of infection. In other environments, these same pathogens might produce polycyclic epidemics. Diseases caused by soil-borne plant pathogens. Many of the root rots, vascular wilts and other diseases caused by soil-borne pathogens also produce only one infection cycle per crop cycle. The inoculum generally is some kind of survival structure resistant to desiccation or freezing, such as Sclerotia, Chalmidospores or oospores in the soil or mycelium in crop residues. This inoculum is dispersed in the soil by plowing and fitting the land and turning under the crop residues. As the roots of the newly planted crop grow through the soil, they encounter the propagules of the pathogen embedded in the soil matrix and become infected. The epidemic progresses as new infections occur, but since any new inoculum that is produced is not dispersed until the soil is again cultivated, there is only one complete infection cycle per cropping cycle. To be sure, not all soil-borne plant pathogens produce monocyclic epidemics, and one must be very careful to understand the life cycle of each pathogen before drawing conclusions about its epidemiology. Demicyclic rusts. Some rusts produce no uridinospore stage, that is, repeating stage on a single host, and the inoculum produced on one host species generally must infect a different host species. This alteration of hosts appear to have evolved in adaptation to the annual growth cycles of the hosts, and we see one infection cycle on each host per year. An example is the cedar apple rust, where all the inoculum that infects apples comes from galls on red cedars, and all the inoculum that infects red cedars comes from apple leaves and fruit. The epidemic on apples occurred during a 4 to 6 week period of the basidiospore production in the spring. A second monocyclic epidemic on red cedars occurs during a brief period of ariospore production in late summer. Let us now look at in detail about the polycyclic epidemics. In order for an epidemic to be considered polycyclic, there must be repeated complete infection cycles, that is, infection followed by pathogen development, new inoculum production, dispersal to new susceptible sites and new infections, all within a single crop cycle. A good example is the potato late blight, where a single cycle of infection, lesion development, sporulation, sporangium dispersal and new infection can occur in as little as 5 days, and many overlapping cycles occur simultaneously during periods of favorable weather. Each cycle can produce more than a 10-fold increase in numbers of sporangia landing on susceptible sites and the explosive epidemic results. Cereal rusts behave similarly. A single uridinospore can infect to produce a pustule from which hundreds of new uridinospores can be released to infect and produce hundreds of new pustules repeatedly throughout the season. Most plant diseases caused by bacteria are polycyclic, and many plant viruses with the aid of insect vectors also can produce repeated cycles of infection in one season. Let us now look at combinations of monocyclic and polycyclic epidemics. Not every plant disease epidemic is clearly either monocyclic or polycyclic. Epidemics produced by fungi with two spore stages can have elements of both, sometimes in distinct phases and sometimes occurring simultaneously. For example, fungus Venturaria inaqualis, causal agent of apple scab. The symptoms are generally most noticeable and serious on leaves and fruit. The first lesions seen in spring are usually on the underside of expanding leaves. Once the leaf opens, the upper surfaces also become vulnerable to infection. A lesion first appears as an area which is lighter shade of green than the surrounding leaf. The lesion is usually circular, and as it increases, the size becomes olive colored and velvety due to the production of asexual spores. Lesions that form on expanded leaves are usually smaller because older leaves are more resistant to infection. Affected tissues eventually may become distorted and puckered, and the leaf lesions often become cracked and torn. Lesions on the leaves and fruit are generally blistered and scabby in appearance, with a distinct margin produces ascospores on the dead, infected leaves that have overwintered from the previous season. These ascospores are released over a period of 6 to 8 weeks in the spring and infect the newly expanding apple leaves. Since no new ascospores are produced until the following spring, this component of the epidemic might not be considered monocyclic. However, each leaf lesion within about 10 days produces a second type of spores, conidia, that also can infect newly expanding leaves. Thus, for the early part of the season, a polycyclic epidemic is superimposed on a monocyclic epidemic. Since the lesions produced by conidia cannot be distinguished from those produced by ascospores, the net effect appears to be a rapidly growing polycyclic epidemic. The epidemic of tan spot on wheat caused by Phyrenephora triceti repentis also comprises both polycyclic and monocyclic elements. The initial inoculum occurs on the infected seed, and the epidemic starts as these seeds germinate to yield seedlings bearing leaf lesions. Conidia are disseminated by wind and splashing rain in repeated cycles of leaf infection throughout the period of crop development. This is by the polycyclic phase. By the time flowering begins, the inoculum for the second phase of the epidemic has built up on the upper leaves of the plant, principally the flag leaf. The flowers and developing grains are susceptible for a relatively brief period to infection by the inoculum extent at the time of flowering, and the lesions on the glumes of developing grains do not produce inoculum capable of infecting more grains. Seed infection, therefore, is a monocyclic phenomenon. It is these infected seeds that provide a major means of carryover of the pathogen from one crop to the next and over the initial inoculum for the new crop. With this, we come to the end of the plant disease epidemiology. Thank you.